Hi, and welcome to my chapter 16 through 19 podcast on classification. First things first, a couple of students asked me to give some shout outs to them, so I have to give a shout out to Steph and Jules. Thanks for watching. So let's go. What is classification? It's essentially figuring out how things should be named and where they all fit together. Originally, we would just look at things and look at what they're like, and then we would give it a name. And so things like mushrooms are actually classified as plants. But now what we're trying to do in our classification systems is to have that classification fit phylogeny, in other words, evolutionary family tree. So let's go. This is the first phylogenetic tree of life, and so if we looked over here, you can see that there are three main branches. The first branch is going to be that of the bacteria. We then have another branch which split into the archaebacteria and then eukarya over here. Remember, we used to classify bacteria and archaebacteria into one big kingdom called Monerin, but now we've just delineated that, and so each of those get a different domain. On this chart over here on the side, I've tried to compare some of the bacteria, archaea, and eukarya as far as some of the characteristics. And if you look at like ribosomal RNA sequences, RNA polymerase, introns, remember, and those are sections of the genes that don't code for a protein. If you look down the way here, peptidoglycan in the cell walls, histone around the DNA, what you find is that our cells, those of eukarya, are actually much more similar to those of archaebacteria. So even though they don't look like us, they're more related to us. What's the biggest piece of evidence? Just looking at that DNA, so sequencing it. So let's start with bacteria. Bacteria are classified according to their shape, and so these are cocci. They come in three shapes. They can be spherical, we call that cocci. They can be spiraled or spirali, or they can be hot dog shaped, and that's like a bacilli. These right here are Staphylococcus, and so if you've ever heard of like getting staph food infection, that's a coccus type of bacteria. This is Spirili. A lot of the sexually transmitted diseases will actually bore into your body, and that's one of the reasons they have that spiral shape. And then this is bacilli. A really good example of bacilli would be E. coli, Escherichia coli, or E. coli. Some characteristics of them, they have this gelatinous capsule around the outside. It kind of protects them. Remember, they still have a cell membrane, cell wall on the inside, but around the outside, some of them have a capsule. It allows them to attach on. They also have these pili, which are like little hairs, and so they can grab onto a surface. So, for example, there are literally billions and billions of bacteria on my hand, and it's the capsules and the pili that are adhering them to my skin. They also have flagella. Flagella are going to be these whip-like structures that allow them to move around, and they move really fast, bacteria, in relation to their body size. And then another cool characteristics of bacteria is that they can form endospores. In other words, when conditions aren't good, they can essentially take their DNA and store it in a spore. And so we heard in the news a few years ago about anthrax being mailed in the mail. Those were endospores. And so what they were doing was loading up some endospores in an envelope, and then when you open it up, and if you breathe that in, then it can activate them, and those bacteria can start living inside your body and feeding on your lungs, which is not good. Next big group, I've kind of skipped over archaebacteria. Remember, archaebacteria live kind of in the extreme areas like Yellowstone Park or in salt marshes, in like high methane areas or really salty water, or in the oceans. Next big group are the protists. Protist is not a real good term. It's a general term, but protists are eukaryotic, so they have a nucleus. They also have like membrane-bound organelles, but they're usually single or very simple in structure. But again, it's like the junk drawer in your kitchen. It has everything in it that scientists don't know where to classify it. Two big characteristics, though, are endosymbiosis. Remember, that was developed by Lynn Margulis. It's this idea that mitochondria and chloroplast were once cells on their own, and then they formed these symbiotic relationships with cells, and then they eventually became part of that cell. Here's some examples, like this is a amoeba right here eating a paramecium. This is stentor, Chlamydomonas. This is foraminifera. But these are all protists, and so you can see even some of those structures inside that protist. The other big characteristic is that of multicellularity. That's the idea that we had a few of these cells that were living together. They formed colonies, and eventually those colonies became multicellular organisms. So this is Volvox, one of my favorite protists. They'll have these little daughter colonies inside the colony, and then when it breaks open, each of those will form a new colony. So you can kind of imagine how that was maybe a number of cells living together and eventually became one organism. Okay, let's keep going. Plant evolution as we branch off from that. And I've covered this in my plant podcast, but if we look at the evolution of plants, there's been 4 major changes. Number 1, they moved onto land. Number 2, they developed vascular tissue, and so vascular tissue is going to be found like in the ferns, so they were able to move material up higher, and that allowed them to get bigger. Next thing is the arrival of a seed. Seed allows them to transport their genetic material far away and prevents it from desiccating, so drying out. And then the last thing is the arrival of the flower. And so each of those are delineated by bryophytes, moved onto land. Ferns have vascular tissue. Gymnosperms have a seed, but it's a naked seed. And angiosperms eventually have the flower. But remember, those flowers are split into 2 groups, the dicotyledons and the monocotyledons. Next are the fungi. Fungi have a really kind of a crazy lifestyle. What they can do is that they can produce haploid spores, which can form new mushrooms, or they can reproduce sexually to produce more haploid spores that will eventually grow. Another cool thing that they have is this idea where one hyphae can grow into another hyphae. Each of those have a nuclei, and now we have a dikaryotic cell. In other words, it has 2 nuclei inside it. Fungal evolution is pretty simple. We've got chytrids. Those are not fungi, but they're animal fungi-like, and that one of the reasons why we think that we're much more related to fungi than plants. We then have the zygomycota. Zygomycetes are going to have the zygosporangia, where they produce the spores. Then we have the glomerulocytes, which are going to be symbiotic relationships that live with plants, and they're really important in mycorrhizal relationships. We then have the ascomycota. Those are going to produce their spores in an ascus, and then the basidiomycota, which are going to produce theirs on a club-like basidium, so it kind of looks like this, and then they have 4 spores that hang off of that. Next, animals. What are animals? Well, they are multicellular movable heterotrophs. That means they're made of more than one cell, so that's why we rule out like a paramecium. Have to be able to move around, and then they're heterotrophic, so they can't make their own food. Almost all of the major phylums of animals that we have today evolved during what's called the Cambrian explosion. So we had this explosion in the fossil record, and descendants of each of those groups are still found with us today. Okay, let's go through animal evolution. I just got a couple of minutes, but it's important to talk about this. There are, again, some breakpoints in this. First group are going to be what are called the sponges, and they're sponges because they don't have true tissues. And when I say tissues, I mean they don't have an endoderm, mesoderm, ectoderm. So that's the first branch. And so sponges doesn't look like an animal, but it clearly is. Next group we have tissues, but they show radial symmetry. So an example of this would be like a jellyfish, which floats like this, or like a sea anemone, which sits like this. And they're very closely related. They've got one mouth, and that also forms their anus as well. Next branching off is going to be the arrival of the bilateral symmetry. In other words, instead of being radially symmetrical, they're actually, you could cut them right down the middle. Next thing is this big delineation between what are called the deuterostomes and the protostomes. And that's when you're undergoing this gastrulation, there's a pore in there, and if that pore becomes your mouth, then you're what's called a protostome, and if not, then you're called a deuterostome. And so we have these big groups. Again, you want to learn each of these, from the flatworm to the mollusks like a snail and all the way up to an octopus. The annelids are going to be like earthworms. Those are segmented worms. We've got arthropods. Those are going to be like the crustaceans, the insects. And then finally we have the nematodes, which are incredibly common, and those are simple worms as well. But who are we related to? Chordates. We're much more related to echinoderms. I've got 50 seconds left. Echinoderms, even though they're radially symmetrical, they actually came back to that. Okay, last one then is the arrival of vertebrates. Vertebrate evolution, if we look at those classifications,